Biomolecular structure is the intricate folded, three-dimensional shape that is formed by a molecule of protein, DNA, or RNA, and that is important to its function. The structure of these molecules may be considered at any of several length scales ranging from the level of individual atoms to the relationships among entire protein subunits. This useful distinction among scales is often expressed as a decomposition of molecular structure into four levels: primary, secondary, tertiary, and quaternary. The scaffold for this multiscale organization of the molecule arises at the secondary level, where the fundamental structural elements are the molecule's various hydrogen bonds. This leads to several recognizable domains of protein structure and nucleic acid structure, including such secondary structure features as alpha helixes and beta sheets for proteins, and hairpin loops, bulges, and internal loops for nucleic acids. The terms primary, secondary, tertiary, and quaternary structure were introduced by Kai Ulrich Linderstrom Lang in his 1951 Lane Medical Lectures at Stanford University. The primary structure of a biopolymer is the exact specification of its atomic composition and the chemical bonds connecting those atoms, including stereochemistry. For a typical unbranched, uncross-linked biopolymer, such as a molecule of a typical intracellular protein, or of DNA or RNA, the primary structure is equivalent to specifying the sequence of its monomeric subunits, such as peptides or nucleotides. Primary structure is sometimes mistakenly termed primary sequence, but there is no such term, as well as no parallel concept of secondary or tertiary sequence. By convention, the primary structure of a protein is reported starting from the amino terminal N to the carboxyl terminal C end, while the primary structure of DNA or RNA molecule is reported from the 5' end to the 3' end. The primary structure of a nucleic acid molecule refers to the exact sequence of nucleotides that comprise the whole molecule. Often, the primary structure encodes sequence motifs that are of functional importance. Some examples of such motifs are the C/D and H/ACA boxes of Snornas, LSM binding site found in spliceosome RNAs such as U1, U2, U4, U5, U6, U12 and U3, the Shine-Dalgarno sequence, the Kozak consensus sequence, and the RNA polymerase III terminator. The secondary structure is the pattern of hydrogen bonds in a biopolymer. These determine the general three-dimensional form of local segments of the biopolymers, but does not describe the global structure of specific atomic positions in three-dimensional space, which are considered to be tertiary structure. Secondary structure is formally defined by the hydrogen bonds of the biopolymer, as observed in an atomic resolution structure. In proteins, the secondary structure is defined by patterns of hydrogen bonds between backbone amide and carboxyl groups. Side A chain main chain and side A chain hydrogen bonds are irrelevant, where the DSSP definition of a hydrogen bond is used. In nucleic acids, the secondary structure is defined by the hydrogen bonding between the nitrogenous bases. For proteins, however, the hydrogen bonding is correlated with other structural features, which has given rise to less formal definitions of secondary structure. For example, in general residues in protein, helices adopt backbone dihedral angles in some region of the Ramachandran plot. Thus, a segment of residues with such dihedral angles is often called a helix, regardless of whether it has the correct hydrogen bonds. Many other less formal definitions have been proposed, often applying concepts from the differential geometry of curves, such as curvature and torsion. Structural biologists solving a new atomic resolution structure will sometimes assign its secondary structure by eye and record their assignments in the corresponding protein databank (PDB) file. The secondary structure of a nucleic acid molecule refers to the base pairing interactions within one molecule or set of interacting molecules. The secondary structure of biological RNAs can often be uniquely decomposed into stems and loops. Often, these elements or combinations of them can be further classified, for example tetraloops, pseudoknots and stem loops. There are many secondary structure elements of functional importance to biological RNAs. Famous examples include the row-independent terminator stem loops and the transfer RNA, TRNA, cloverleaf. There is a minor industry of researchers attempting to determine the secondary structure of RNA molecules. Approaches include both experimental and computational methods. See also the list of RNA structure prediction software. The tertiary structure of a protein or any other macromolecule is its three-dimensional structure, as defined by the atomic coordinates. Proteins and nucleic acids fold into complex three-dimensional structures which result in the molecule's functions. While such structures are diverse and complex, they are often composed of recurring, recognizable tertiary structure motifs and domains that serve as molecular building blocks. Tertiary structure is considered to be largely determined by the biomolecule's primary structure, its sequence of amino acids or nucleotides. The quaternary structure refers to the number and arrangement of multiple protein molecules in a multi-subunit complex. For nucleic acids, the term is less common, but can refer to the higher-level organization of DNA in chromatin, including its interactions with histones, or to the interactions between separate RNA units in the ribosome or spliceosome.